This is the Professor, CBMD PhD, and today is Wednesday, August 5th, 2026. It's 8:25 and 40 seconds, p.m. Eastern Standard Time, and today's research, jumping genes. So let's hop to it here. The researchers and scientific investigators catch a jumping gene, mid-leap between species. Date August 4th, 2026. Source, Max Planck Institute for Marine Biology. Scientists have uncovered a surprising way that jumping genes may move between species and speed up evolution. While these mobile genetic elements were thought to travel mainly inside viruses or plasmids, researchers found circular intron, RNA from a tiny predatory bacterium inside the dead cells of another microorganism. Because the RNA forms a stable ring that resists breakdown, it may provide jumping genes with a previously unknown route for spreading between species. Jumping genes are genetic parasites found in bacteria, plants, animals, and humans. They can be released inside cells as small RNA molecules from ribonucleic acid, RNA, then use specialized mechanisms to insert themselves into new locations within the genetic material. These movements can give cells new traits, making jumping genes an important force in evolutionary change. Some jumping genes can also remove themselves from RNA with the help of an RNA enzyme. These elements, known as ribosomes or self-splicing introns, represent a distinctive group of mobile genes. Moving within a single cell is one thing. Crossing into another cell, or even another species, is far more difficult. Genetic family tree studies show that such transfers have occurred, but scientists have generally assumed that jumping genes traveled as passengers inside plasmids or viruses. Researchers have now observed something unexpected that points to another possible route. The researchers studied a slowly growing enrichment of bacteria and archaea that produces methane biogas. The community contained an unusual dominant organism, a very small predatory bacterium, Candidus velominococcus archaeovorus, feeds on microorganisms that convert limestone, the compound responsible for the scent of oranges, into methane and carbon dioxide. Within the filaments of methyltrypsinogen, the most important methane producer on Earth, the researchers noticed that individual cells were dead. They suspected that Candidus velominococcus archaeovorus might be responsible. To test that possibility, they needed to find molecules from the predator inside the dead cells. While examining the genome of Candidus velominococcus archaeovorus, researchers identified an intron that functions as a jumping gene. Intron RNA had never been detected outside a cell, which made the researchers especially interested in looking for it inside the bacterium's prey. The researchers and scientific investigators from marine microbiology have developed methods sensitive enough to detect very small amounts of RNA in bacterial cells. Using specially designed nucleic acid probes, the team produced microscopic images showing intron RNA in living cells of Candidus velominococcus archaeovorus in the dead cells of Mathematician. The researchers had effectively caught the intron while it was trying to replicate. However, Candidus velominococcus archaeovorus had already killed a new host. The gene's attempted transfer therefore ended as a jump into an empty cell. Ribonucleic acids act as messengers in living cells. These long-chain molecules carry instructions from the genetic material to the cell's protein factories. They are normally broken down quickly, beginning at their exposed ends. For that reason, dead cells usually do not contain ribonucleic acids. The intron RNA survives because it forms a circular molecule with no open ends. The ring-shaped structure protects it from enzymes that would otherwise break it apart. The stability of intron RNA and its ring form a distinctive feature. In humans, circular RNA molecules influence many metabolic processes, and their role in tumor development is currently the subject of intensive research. Applications in RNA vaccines, for example, against the COVID virus and certain forms of cancer, are also in the pipeline. Our study showed that in microorganisms, jumping genes can be transferred to other species via their circular RNA. And this videography was created for educational purposes only. My code name is Will. I'm Professor Christopher Brunet, leader of research scientist, ufologist, psychic, computer technician, computer programmer, philanthropist, writer, mathematician, artist, photographer, film producer, chef, animal, and nature lover. My passion is world peace. Have a great day.